So today we're going to look at DNA structure and nucleotide structure for lower sixth. This is a lower sixth topic. So these are the objectives, as per usual. They're at the top of the screen. And the first thing we're going to look at is the properties of the genetic material. So number one, properties. Properties of the genetic material. Now, this essentially is just looking at whatever the genetic material is, and of course, not that long ago we didn't actually know that it was DNA. We just knew that it had to somehow encode information and be passed down from generation to generation. But we did know that three things had to be true. Okay, number one is faithful replication. Okay, in other words, we have to have some mechanism by which the genetic material is replicated. The second thing is that it needs to be stable, specifically, obviously, at physiological temperature as well. And therefore, the whole idea of that, of course, is that it doesn't disintegrate over time, or certainly very quickly. And it's not prone to being degraded at all. The final thing is it must contain coding capacity, right? If it's going to act as a genetic code, of course, it has to have some kind of ability within it to encode information to make proteins. So those are the three things that DNA obviously has. We now know it's DNA, but back in the day, people thought that it was potentially proteins and that kind of thing. So that's hopefully objective number one, nice and easy, fairly quick for that. Number two is to look at what a nucleotide looks like. So number two, nucleotides. And for this, you hopefully are aware from GCSE that the monomers, so the monomer of DNA and RNA are things called nucleotides. Okay, so these act as kind of like the sort of Lego bricks, if you like. And these form a polymer, so we can join them together here in a repeating fashion to form a polymer of nucleotides. Okay, and hopefully what you are aware of is that we have two types of polymer. So these are called polynucleotides. Polynucleotides, and we have DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. And these are obviously, as the name suggests, quite chemically similar to each other. So what we're going to do is look at one of these individual building blocks. So if I zoom into one of these nucleotides, what do they look like? And it consists of three things. The first is a phosphate group, and it's inorganic phosphate, so we represent that as Pi for phosphate. Number two is we have a pentose sugar, and hopefully you remember what pentose means, obviously five carbons, and it's one of two types, depending on whether you're DNA or RNA. It will either be ribose or deoxyribose. Okay, and just to sort of ruin the surprise now, DNA contains deoxyribose, so this one is quite common in DNA, and RNA uses just ribose. Okay, so just make sure you're happy with that. Of course, DNA starts with a D, as does deoxyribose, and ribose an R, and RNA begins with an R. So hopefully fairly straightforward on that. The final thing is a organic nitrogenous base. Okay, and we've got different flavors of those as well. which I'll talk about in a minute. So all of these things, these three things come together to form a nucleotide. So let's draw them out. So number one is the phosphate group. Typically, we represent it as pi, so I might do that in future. But in terms of what it actually looks like, it looks like this. So I'm going to draw it down here. So phosphate, and you need to know the structure. Oops. Looks like this. And specifically, this is not phosphate yet. This is called phosphoric acid. Okay, it is a weak acid, and as a result, it will donate some of its protons. It actually donates 2 of them. So we can see that here. So I'm going to get rid of this guy and this guy here. In fact, just for ease, I'll make it these 2. And that gives these both a negative charge. Okay, this is now not phosphoric acid. This is now something we call phosphate. 8 being or referring to the conjugate base of the acid. So that is, this here is now phosphate as opposed to phosphoric acid. It was once phosphoric acid, but is no longer. So I'm going to move that up here a little bit, just to give me a bit of space. Okay. What then happens is, hopefully you are aware, or you're looking at this and thinking, this OH group here acts as a nucleophile. And that is, of course, because the oxygen here has 2 sets of lone pairs, here and here, which can do nucleophilic attack. So I am going to just get rid of that. We're going to get rid of this hydrogen and draw to the next molecule. So that oxygen here will do nucleophilic attack and can form a bond with a sugar. And the sugar is a pentose sugar, so meaning it's made up of 5 carbons. And it's going to either be ribose or deoxyribose. Okay, so we're going to start with ribose. So we're going to come along here, down, and I'm just doing sort of mainly skeletal formulae for this. Oops. Like that. So that is our ribose, which you should be aware of in terms of structure from the carbohydrates lesson. And ribose has 2 OH groups sticking off here. It has hydrogens elsewhere as well, and a couple of other things, but we're not going to worry too much. Just to be clear, where the carbons are, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So I'm just going to make that clear. Carbon 1, 2, 3, 4, and 5. And hopefully you remember from the sugars lesson on carbohydrates, we number them with a prime in DNA or RNA. So it's the 1' carbon, the 2' carbon, etc. The reason why we number them with a prime is because of the final thing, which is that we connect to an organic nitrogenous base. Okay, so you have to say, in fact, I'm going to write that. You have to at least say that it's a nitrogenous base. Okay, it is not a base in terms of, sodium hydroxide or that kind of thing. It is an organic molecule that we call a base. So this base here has carbons within it. Okay, there are 2 structures to this. Okay, so it can be of 2 types essentially. You can either look a bit like this, where it's an organic carbon ring of 6 carbons. There's maybe a couple of other things attached, but we'll get to that in a minute. Or you will have a 6-membered ring connected to a 5-membered ring. So I would say this is like a house and a garage. The house is the 6-membered ring, and then you've got the garage next to it. These are all carbons essentially, and you might have oxygens and nitrogens sticking off, but or as part of the structure. But it's an organic molecule. It's an organic ring. You need to know the names of these, and I always remember this just because the more simple word is the more complicated structure, annoyingly. So purines So purines sounds pure because that's what it starts with, but ironically, it's actually the more complicated of the two structures that have the simple name, because the one up here is called a pyrimidine, or pyrimidines have the structure of just a 6-membered ring. The only other thing you need to know is a mnemonic, which I call PUGA, like that, which stands for purines are G and A. So guanine and adenine as two of your 4 bases in DNA. G and A are your purines, Pu. So G and A, guanine and adenine, are down here. They will have this double ring structure, whereas the rest have the single ring structure and are referred to as pyrimidines. So this would be T for thymine and cytosine, and uracil if you're talking about RNA. So this is in RNA. Hopefully we know that from GCSE though. That's all you need to be able to distinguish between the two. You don't need to know any more sort of chemical detail, but I will show you in a minute. So that is what your nucleotide looks like. And just to point out, going back to this sugar in the middle here, you've got 2 types, okay? This is ribose, okay? So ribose is just the standard sugar as I've drawn it here. If you are deoxyribose, okay, which is the sugar that DNA uses, okay, so DNA nucleotides use deoxyribose. Hopefully what you can appreciate is deoxy means lacking oxygen, and I always teach my students to know that the actual name of this is 2' deoxyribose, because it tells you exactly which of these carbons needs to be removed. So if this was a DNA nucleotide, we would need to get rid of, or you know, the sort of cell gets rid of when it uses these molecules, that oxygen there on the 2' carbon. Okay, that actually makes this molecule very, very stable. Okay, so 2' deoxyribose is much more stable, increases stability. Oops. Compared to ribose. Okay, so this actually is a much better thing for the cell to do, and the reason is because this oxygen here, this OH group, acts as a nucleophile, and therefore if you get rid of it, we are less prone to or to doing nucleophilic attack and sort of causing hydrolysis of your nucleotide. The only other thing left to say is, just to be clear, the 3' carbon is left intact, and this nucleotide will now be used to join on to other nucleotides to form a dinucleotide, then a trinucleotide, and then eventually a polynucleotide. So just to zoom into what these look like, here we go. It is worth pointing out again that your purines are the more complicated structure. So here you've got a 6 followed by a 5, and here you've just got a 6. So pyrimidines are the more complicated name, but the more simple structure. And you can see the purines here are G and A. So remember, we remember that by PUGA. Tells us that the purines are G and A, or guanine and adenine. And this is what they look like. Same thing for cytosine, uracil, and thymine. Worth pointing out, of course, that in RNA, we use uracil and not thymine, and yet the only difference between these two, between uracil and thymine, is the methyl group here. Okay, so you can think of thymine, if you like, as 5-methyl-uracil. Okay, it's on the 5th carbon here. So this is, you know, AKA 5-methyl-uracil. And it's not, it's called thymine, but that's essentially what it is. And hopefully you can appreciate, therefore, that the structure is really not that different. So, you know, substituting one for the other in RNA is quite easy. Why exactly that happens, I'm not going to go into, but if you're interested, you can always speak to me. But again, it helps DNA to have thymine as opposed to uracil. The only other thing to point out is that there's a lot of hydrogen bonding potential. And the reason why is because you've got lots of electronegative atoms in the nitrogen here. So all of these things, especially NH here, double bond oxygen, that's electronegative, NH here, all of these that I'm highlighting now, I'll not highlight the methyl group, are very good for hydrogen bonding. So being a hydrogen bond donor or being potential hydrogen bond acceptors. Okay, so you can hopefully see that I've highlighted quite a lot there, and that is because we've got lots of either, in the case of oxygen here, delta negative atoms, because they are electronegative, or you've got electronegative, so delta negative atoms, such as the hydrogen there, directly connected to a hydrogen, making the hydrogen delta positive. Okay, so what this means is we have lots of abilities to form hydrogen bonds. Okay, and you need to know or appreciate the definition of a hydrogen bond, which is that you need a electronegative, or it's when you have an electronegative atom connected or bound to a hydrogen atom, which makes the hydrogen atom, by the way, delta positive, and that is nearby another sufficiently delta negative atom. Okay, and typically we refer to that as either fluorine, oxygen, or nitrogen as the major electronegatives. So this is shown here, and actually because of the specific shapes of these bases, these organic nitrogenous bases, they can form hydrogen bonds and specific numbers. So A and T, hopefully you remember from GCSE, can form 2, if I highlight them here, hydrogen bonds. Don't forget, if you're ever going to draw these, you need to draw them with dotted arrows to show that they are intermolecular forces. Between G and C, we have 3. And it's also worth just pointing out that thymine here, of course, can substitute for uracil as well. So 3 hydrogen bonds between G's and C's, and 2 hydrogen bonds between A's and T's, or A's and uracils. So worth bearing in mind that. Right, moving on. So that is the nucleotide. There we go. We now go to sort of naming nucleotides. So I just want to do a little bit of, or give you a little bit of information on this. So naming. And you need to know 2 things, which is that the nucleotide, I remember this as phosphate. Okay, so nucleotide has a T in it, as does phosphate. So what a nucleotide is it's a sugar and a base with inorganic phosphate attached, okay, as we've seen. Okay, and the nucleotides can be named because there's something called a nucleoside. And I remember this because S stands for sugar. So this is quite literally just the sugar and the base. No phosphate. Okay, so there's no T, so no phosphate. And you name a nucleotide because you name the nucleoside plus the number of phosphates attached. So it's a bit complicated, but I'll give you an example. So you need to know that the nucleotides, in fact, the bases, the organic bases, are adenine, thymine, both forming 2 hydrogen bonds, guanine, and cytosine. And we're going to put uracil in as well. There we go. Those are the bases. If we now look at the nucleoside, which is going to be the base plus the sugar, okay, if we don't say anything else, we just assume it's a sugar. It's assumed to be the plain normal kind of ribose sugar, not the 2' deoxy. So adenine, then we call it adenosine. Okay, so adenosine means we've got adenine as a base, and that is connected to a ribose sugar. So we 2 hydroxyl groups, so it's not 2' deoxy there. Okay, so it is the ribose sugar here plus adenine as the nitrogenous base. Okay, so that whole structure there would be called adenosine. If DNA version where we use 2' deoxy, we call it deoxy adenosine. Okay, we add the deoxy prefix. So thymine becomes thymidine. Guanine becomes guanosine. Cytosine becomes cytidine. And uracil becomes uridine. Okay. So again, all of these are the ribose sugar here connected to one of the bases, either A, T, G, C, or U, depending on what we call it. If I were to just refer to adenine, I am talking only about the base adenine, not the adenine connected to a sugar, essentially. So again, side for sugar, nucleoside for sugar. Okay. Now, what you can then do is add phosphates to make the nucleotide. And to name the nucleotide, we name the nucleoside, and then we state the number of phosphate groups that we're adding. Okay, so adenosine with 1 phosphate group attached. So don't forget, adenosine is this structure. I've got adenine connected to my ribose sugar. If I'm going to simply add 1 phosphate group to it, in fact, that's poorly drawn, it should be like that, that's ribose. So if I'm going to add my phosphate group to it here, it would be adenosine mono for 1 phosphate. If I was to add 2, it would become diphosphate. If I added 3, it would become tri or tetra if it's 4, etc. Okay, and I point this out because you have already come across one of these. Adenosine triphosphate looks like this, where I have 3 phosphates connected to a ribose sugar. Oops, that's poorly drawn. Connected to adenine as the base. This is ATP. Adenosine triphosphate. Okay, adenosine being this bit, so that's adenosine. And then this is adenosine triphosphate. There we go. Oops. And just the adenine there, the base on its own is just adenine. That's the base. Okay. I'm not going to do that for every single one because that's the main one that you need to know, but it's worth bearing in mind that you can have guanosine triphosphate or cytidine triphosphate, etc. Or you could have guanosine diphosphate. They all exist. So of course, ATP is the energy currency of the cell. It's the high energy molecule that is used for liberating energy when we hydrolyze these phosphor anhydride bonds that connect the phosphates. These are high in energy. So that's how you name nucleotides. And again, just to be absolutely clear, because I just call it adenosine triphosphate, oops, Okay, this is the ribonucleoside technically, because it's not deoxyribose, the sugar is ribose. If it was deoxyribose, I would call it deoxy triphosphate. But I don't do that. So ATP, as we know and love, adenosine triphosphate, is a ribonucleoside triphosphate. Okay, because it is using ribose as the sugar, not deoxyribose. So hopefully that makes sense. So that's naming nucleotides. So again, name the nucleoside first, and then how many phosphate groups are attached. And if it's deoxy, we need to put the word deoxy in front of the nucleotide name. Okay. The next thing is condensation reaction. So option number 4 is condensation reaction. Putting nucleotides together, essentially. So, of course, we've looked at one of these building blocks for DNA, but DNA and RNA, in fact, are both polymers of nucleotides. Therefore, we need to know how to put them together. So, let's just assume I've got a DNA chain. So, I've got my phosphate here. In fact, I'm going to do it more simply first. I'll just draw it like that, I think, for now. And I'm going to draw my, if this is in DNA, it's deoxyribose. So I'm going to put my OH on the 3', but I'm going to leave the hydrogen here because it's deoxyribose. And I've got my base, so whatever that base is. What happens for the condensation reaction is you need a substrate that can react with this. You need another nucleotide here. Let's say it's got a G. Another DNA nucleotide, so it's got another, it's deoxy again, so 2' deoxy, don't forget. 2' deoxy. And this is going to act as a substrate for essentially replication, so adding nucleotides to form a polynucleotide chain. And for reasons that I'm not going to bore you with too much, apart from I'll briefly give you an idea, but I'm not going to go into the super detail. You need to know that this oxygen up here is a nucleophile. It has 2 sets of lone pairs, and it will therefore act to do nucleophilic attack. And if you were to zoom into this phosphate group, of course, it would look like this. Oops. Okay, and the key thing is that the phosphorus is surrounded by delta negative oxygens. So the phosphorus in the middle here will be delta positive, acting as a good electrophile. So the mechanism for this, if I do it in red, would be that one of these lone pairs attacks this this phosphorus atom in the middle, and then one of these would then break off. Okay, to form the most stable leaving group which would be the thing that's connected here. Okay, so that would break off there. Now, you don't have to worry too much about that, apart from to say that that's why I'm going to draw this arrow like this. Okay, it's attacking the phosphorus. And we therefore, if we form a bond, now need to break a bond. And the only bond that we have available would be this one. Oops. And it would break like that. But the problem with that, of course, is that we would then break the phosphate off the nucleotide. Okay, so all I'd end up doing would be sticking a phosphate group onto here, and the sugar, essentially the nucleoside, so the sugar and my next base would just diffuse away and not be used in my DNA molecule, my polymer. So we can't do that. Okay, and there are other reasons as to why, but that's the main one, right? Which is basically to say that this molecule, as I have drawn it, is not a substrate for adding on to a chain of nucleotides or undergoing a condensation reaction. it turns out that this nucleotide here, which I'll draw like this, has to be activated. Okay, so it has to be activated. So not only does it have to be a nucleotide, we have to be very specific. It's not just a nucleotide, in this case monophosphate, or if I'm being technically accurate, it's a nucleoside, which is this bit, monophosphate. It has to be a triphosphorylated nucleotide. So we call it a nucleoside triphosphate. Because we're naming the phosphate, we're saying that it's actually a nucleotide. So sometimes you'll see it written instead of nucleoside, we can absolutely still call it a nucleotide because it does have a phosphate, at least one of them there. So it's a nucleotide triphosphate. So I'm now going to draw two other phosphates there. And that is a substrate for nucleophilic attack. Okay, so what will happen if I change color is my oxygen here will attack this phosphorus in the middle of this phosphate group, and I can now break off this bond here. So those electrons are given back to that phosphorus, and essentially we're sort of cutting that bond there. And what you end up with then is the leaving group, which is these two phosphates. Okay, and we've connected this oxygen now to the phosphate on the next nucleotide. So the product would look like this. So I've got my nucleotide that was up top, which had an adenine on it. We then have my oxygen here, and then that connects straight to my next phosphate group, which had my guanine in it. Okay, drawing the hydrogens on there like that. So that's what it looks like. Okay, what I've done is I have formed this bond here. Okay. Now, what that exactly looks like, I will show you in a little bit. But it's worth pointing out this is now a dinucleotide, and the leaving group, these two phosphates, are now over here that are connected together. This is our leaving group. It is called pyrophosphate. So pyrophosphate, that's the leaving group. It is not water in this case. So, you know, it's just worth bearing in mind that condensation reactions are not always liberating water. In this case, they're actually liberating two phosphate groups stuck together. So they're called pyrophosphate. Okay. In terms of what this actually achieves, I'm just going to draw it again, but I think in a bit more detail. So let's imagine a phosphate group here. And I'm going to draw my sugar. It's deoxyribose. So there we go. There we go. Okay, so that is what my nucleotide looks like, and I've just drawn a few more carbons on there. I'm going to draw the same thing again. Okay, for my next nucleotide. So here we go. This is my triphosphorylated deoxynucleotide, deoxyribonucleotide, in fact. And what the mechanism is as follows, where my lone pair here attacks that phosphorus, and this bond then breaks and gets given back to that oxygen. Okay. So if you imagine the two lone pairs of electrons on that oxygen that have just done the attack in green, and I've got two lone pairs in red. Ooh, that's not what I wanted. Two lone pairs in red here that are then going to be broken and given back to that oxygen. Okay, so the end product would be that the There we go. So whatever nucleotide that is, we then have my oxygen, which has formed the green bond here to my phosphate here. That's then given electrons back. That's a carbon with 2 hydrogens on it, don't forget, like that. Okay, so that essentially that green bond is these 2 lone pairs of electrons here that have now essentially formed a bond to that phosphorus, which is that bond there. What was my other point? Oh yes, and of course the bond that's broken. So this purple one here, those electrons have been given back. Those red ones have been given back to pyrophosphate or to 2 phosphate groups. So my leaving group now looks like that, with 2 lone pairs now attached, and therefore that makes that oxygen negative as well. Okay, probably chose the wrong pen for that. Let's go an actual pen. There we go. So those 2 electrons that were here have now been given back to that oxygen, which is there. Now, what we have now formed, and we're nearly there for this part of the video, is this, which is a diester linkage, because you've got your ester group. Oops, you've got an ester linkage here with that oxygen and an ester linkage there with that oxygen there. So it's a diester. And because it's 2 esters with a phosphate group in the middle, or phosphorus, it's called a phosphodiester bond. Okay, so that whole thing is called a phosphodiester bond. The only last thing to mention with this is that there's directionality here. Okay, so I'm going to use this one as an example, I think. So if we're numbering the carbons in the sugar, don't forget this is our 1', 2', because it's 2' deoxyribose, don't forget 3', 4', and 5'. Same thing here. Okay. And what's important is when we link nucleotides together, the whole structure is polar, it's directional. Once we've linked these together, we have the 5' carbon up here connected to the phosphate, okay? And we link, essentially go through like this, and we link to the 3' carbon to the next 5' phosphate, like that. So this is 3' at the bottom, then we go to 5', and then 3', etc. Okay, so essentially when we're joining nucleotides, the whole DNA structure, if I were to very quickly sketch what one would look like, like that. Okay, we are connecting off this end, we've got the 5' phosphate group sticking off, and at the very other end, don't forget we've got a hydroxyl there. It's a 3' here hydroxyl group. Okay, so the whole DNA strand, if you think about it, has a slightly different structure. It's not completely the same. Off one end, you've got a phosphate sticking off, and the other end, once you've finished adding all the nucleotides you need to, you've got a 3' hydroxyl end sticking off. Okay, so we say that this DNA chain, this polynucleotide strand, has directionality, and it goes from 5' to 3'. And that's really important to get your head around. Okay, so the next thing, once we've done the condensation reaction, and I've kind of touched upon the sort of 5', 3' convention, which, by the way, we will come back to that when we do DNA replication. So don't worry about it too much, but just be aware, once you make a strand of DNA or RNA, there is a sort of directionality to it. Okay, it runs from 5' to 3', i.e. the first nucleotide to go in will have a 5' phosphate sticking off the end. If I just do this very scruffily. And then as we add nucleotides, eventually we end up with one right at the very end, which will have the 3' OH group sticking off. Okay, so we've got the 5' phosphate up top and the 3' OH group at the bottom. Okay, that's all that kind of that I need to get across to you. Okay, final bit of this first kind of lesson on DNA is quite straightforward. It's just to talk about the structure of the double helix. So this is obviously making one strand of DNA, and what we can do, if I go all the way back down here, is from my one strand of DNA that I've made in a 5' to 3' direction here, I can make another strand of DNA, and I can connect them together because I can do hydrogen bonding between the base pairs. So if I draw some bases in, let's put some actual bases in. So let's say that was an adenine. Opposite, I can put a thymine. If this was a cytosine, I can put a guanine. If this was a guanine, I can put a cytosine, etc. Okay. So now I've got to try and draw this upside down. These would be connected here. They would then be connected to. We're good at doing this upside down. Here we go, like that. To the sugars, like that. I'm going to flip my thing around. There we go. That will be easier. And then that will, of course, be connected to my phosphate groups, which I won't draw in their full. Like that. There we go. And of course, we've got hydrogen bonds that can form between them. Okay. So in the case of A's and T's, I can form two hydrogen bonds. So I'm going to draw two dotted lines. C's and G's, I form three. Like that. Okay, and we can keep going. But the key thing is that we've got, if I just get rid of this, just to make it a bit clearer, if I keep it in red, I've got my 5' end of this strand and my 3' end down here. So this strand is going in that direction. But if I look for the same, don't forget this would be an OH group there. On the other strand, I've got my 5' phosphate down here, and I'm going in that direction to my 3' hydroxyl group there. Okay, so these strands in my double helix now are anti-parallel. Okay, anti-parallel, and you need to know that. Of course, this whole structure here, when I draw it, and when you've come across it, okay, we sort of draw it like this as a double helix, maybe with the sort of hydrogen bonds like that. Sometimes you might see the bases stuck out in the middle as well, or rungs of the ladder, but the purple lines here are what we call the sugar phosphate backbone. Sugar phosphate backbone. And that is referring to, if I was going to highlight it here, the phosphates and the sugar here on either side, and the bases stick out in the middle. Okay, so that's the double helix. Now, just to go to some experiments here that you need to know about, you need to go firstly this guy called Chargaff, Erwin Chargaff. Oops, there we go. All he did was very early on, when people were trying to discover the structure of DNA and the genetic material, that kind of thing, is he took DNA from different organisms, different kind of domains of life, in fact, and he was able to work out the percentage composition of A's, T's, G's, and C's. Didn't know the structure of DNA at this point, but he was able to work out the relative percentages via various chemical and chromatography techniques. His major discovery here is that the percentages of A's and T's equaled each other. Okay, were the same or similar. not completely identical, but certainly similar. So, 26.1 to 23.9, 31 to 32. There's exactly the same here in the human liver, but very, very close. And that was replicated throughout all of the different organisms he looked at. The same was true for the C's and G's. Okay, so the C and G percentage was the same or similar. So, 24 and 25, or, 19.5 and 19.9, very close. So the A's and T's and the C's and G's, the ratio was always very close to 1, not always absolutely. But the other important thing was that different organisms have different percentages of A's and T's versus C's and G's. And of course, what I mean by that is that, in humans, for example, we've got about 30% A and T, and we've got about 20% C's and G's. So I'm just trying to look for a good comparison here, but let's go E. coli, it's 26 and 23%, and 24 and 25, so, a little bit different. Archaea aren't on here, but if you were looking at Archaea, there's a species of Archaea called Halopharex vulcanii. The percentage GC composition, it's about 60%. So it's the majority GC. Anyway, so he looked at that, and that's basically all he found out. He didn't quite know what to make of it, apart from just stating the facts, I guess. So more importantly was a bit later on, where this lady, Rosalind Franklin, who is in the lab of Maurice Wilkins at King's College London, she did a lot of this stuff, which was X-ray diffraction. And basically, it involves sort of firing X-rays at a sort of crystallized sample of either protein or DNA. And in this case, they use DNA because they wanted to know the structure. And essentially, you get diffraction of the X-rays depending on what atoms are hit. And of course, that produces, if you can use a photographic film or X-ray film, a image like this. This is called image 51. You can look it up if you want to. And basically, you can look at this picture and work backwards to go, well, what was the original structure of the sample based on what the pattern looks like? And you can think of that like having a chandelier. When you shine light on a chandelier, it produces kind of patterns on the walls. And the patterns produced will differ depending on what the chandelier actually looks like and the structure of it. It's basically that as an idea. I'm not going to go into much detail on it. It's incredibly complicated. We use computers to do it now, but even then it's quite tricky. But this work was really quite important and quite pivotal. So what this told her was that she looked at this picture and fairly quickly saw that the sort of X, oops, I'll just move that. The X-like structure here points to the whole structure being a double helix. So you can see that as the phosphodiester backbone. So phosphodiester backbone is those bits in blue, and the sort of thick bits on the outside here and here represent the bases. And the key thing is that it kind of told her that it was most likely a double helix of some sort. Now, history likes to paint it out that Rosalind Franklin had no idea what this picture meant, and that it took Francis Crick, from the Watson and Crick pair to actually look at it, and he immediately knew it was a double helix. I don't buy into that. Rosalind Franklin was an extremely clever woman, very, very good crystallographer and sort of chemist, physical chemist. I don't believe that she didn't really know what it was, but the story goes that Watson and Crick were also, of course, trying to solve the structure of DNA. So Watson and Crick kind of entered here, having already postulated a wrong model. They then got this data from Rosalind Franklin, had a look at it, saw that it was a double helix, and along with their cardboard model, was able to construct a double helix model, because they took the diffraction data with the Chargaff data Okay, so they actually went back to this guy's work and sort of went, oh, hang on a minute, if it's a double helix and A's and T's are similar in percentage and G's and C's are, then maybe it's a double helix. So double helix, and somehow the A's go with the T's and the C's go with the G's. And they basically just used cardboard cutouts of the bases that we've already seen to put them together and come up with a nice model for the double-stranded structure of DNA. So that's a very brief whistle-stop tour of the double helix structure. Just two more things to point out. When we replicate, we'll see this when we do DNA replication in more detail, but this is kind of what I've said to you in that we've got a nucleotide here, so we've got phosphate, sugar, and the base. When the next one comes in, which is here, which is a substrate because it's got 3 phosphate groups, it's a nucleotide triphosphate or an activated, you can call it. It's an activated nucleotide, DNA nucleotide, in fact. What happens is your 3' OH group, which is sitting here, will attack that phosphorus, and this bond here will break off, liberating the pyrophosphate, which is shown here, and you end up then connecting that whole nucleotide to the new structure via a phosphodiester bond. Okay, so this is that there in yellow is the phosphodiester bond. It's a condensation reaction. We liberate 2 pyrophosphate molecules, not water in this case. And the only thing to point out is this is done by an enzyme, of course. Everything's done by an enzyme when we do DNA replication, but it's called DNA polymerase. Okay, does this reaction. And we'll go into a lot more detail about how it actually does it in the DNA replication section. So back to the top, that's the double helix done. Final thing is just to talk about the stability of the double helix and just look at RNA as well. So I'm going to put here why DNA is good as its job. The answer is it's a double helix. And as Watson and Crick pointed out, this means that it can be replicated semi-conservatively. Oops. Okay, so semi-conservative replication. What that means is because it's a double helix that looks like this, I can separate my 2 strands apart, and each one can act as a template to make a whole new strand there. Okay, so we haven't got proof for that yet. I'll talk about that when we do DNA replication, but that's kind of the model they came up with. At the end of their paper, Watson and Crick said basically that it's not escaped our notice that the structure of DNA that we've sort of come across or postulated, I think they said, tells us exactly how the double helix can be replicated. Okay, we can split the 2 strands apart, each one because of complementary base pairing, acts as a template for the other. Just a note on that, actually, if you're describing the structure of DNA, you always get marks here. You always get marks for saying complementary base pairing. Sorry, my pen's being a bit strange. Complementary base pairing, and when you say that word, which you all please do, you nearly always get a mark for saying that A goes with T, G goes with C. Okay, 2 hydrogen bonds up top for A and T, and 3 hydrogen bonds for G and C. Worth bearing in mind. Right, the next thing as to why the double helix structure is good is because we have the phosphodiester backbone on the outside of the molecule, and the bases are protected in the middle. Okay, so if I was going to draw 1 strand like that and 1 strand like that, that's really bad. Let's try it like this. There we go. It's a little bit better. My phosphodiester backbone is on the outside, and of course on the inside I've got my bases. Oops. I'm not going to draw all of them, but bases that are connected by hydrogen bonds on the inside. It's the bases, right, the A's, the T's, the G's, the C's in DNA that contain the coding information. Okay, so it's worth protecting them, right? On the outside, in the nucleus, don't forget this is, so my double membrane of my nucleus, here we go, right? You know, there's harsh things in the nucleus, right? There's maybe some free radical species. There's sort of maybe some enzymes that want to sort of get at the bases, right? It's not a nice environment, so it kind of protects from nasty things. It will kind of protect the bases, okay, and protect your coding capacity and ability to make proteins. Second is the thing about hydrogen bonding is that, don't forget, many of them are very strong indeed. But if you only have a few of them, they're very weak intermolecular forces. And this is useful for DNA because DNA is very long. Okay, so you have lots of these hydrogen bonds all across, you know, hundreds of thousands of base pairs in each chromosome. Therefore, you cannot simply just pull them apart. Okay, so you can't do that, and it's very stable. It won't fall apart. But that's useful because individually they're very weak. Okay, so what that means is you can separate the DNA double helix if you were to do something like this. Okay, oops, that's not very helpful, is it? There we go. And you were to essentially pull them apart from the end. Okay, in other words, you're going to start at this end, pull that strand there, because you can break each one of these hydrogen bonds individually. So you can pull them apart from one end, essentially, without too much effort. Okay, so again, going back to Watson and Crick and you know, the sort of semi-conservative model of replication, you know, pulling those two strands apart to act as a template for other strands and new DNA synthesis probably isn't that much of a deal, a big deal. But when we've made our new strand, it won't spontaneously just fall apart. Okay, so you need an enzyme for this, which we'll talk about later. Final thing is it's a very large molecule, so it can store lots of A's, T's, G's, and C's. So we've got lots of coding capacity. Okay, so lots of coding capacity there. Right, final thing. And we might have done this in about an hour, I'm guessing, without checking the time. But we need to cover RNA, and this will be done in literally a minute or so. So all you need to know is the differences, and we'll talk more about RNA when we do replication. Transcription and translation. You need to know that it is single-stranded. So single-stranded, as opposed to DNA being a double helix. Okay, so this immediately means it's more unstable. Okay, it's going to get broken up. So there's this single strand here with the bases sticking out like that. These bases are free to be attacked, essentially, to be broken and degraded. And so very quickly you'll end up with, you know, bits of broken RNA. Okay, so it's susceptible to hydrolysis. We'll talk about why that's actually a benefit much later on, but there we go. The other thing, of course, is to say that we have uracil instead of thymine, which we'll get to. That also makes DNA a lot more stable. We'll get to why that is later on. The sugar is ribose, not deoxy, not 2'-deoxy. Okay, so in DNA we use the 2'-deoxyribose sugar. In RNA it's just ribose. And finally, it is a short strand or molecule because instead of DNA, where we replicate the entire chromosome, so if this was a chromosome, okay, when we do DNA synthesis, we replicate the whole thing, and DNA contains loads of genes. I'm just drawing 3 on each. side for now, but there'll be loads. Of course, RNA is made only from one gene, maybe this one, okay, where we make just a copy of that particular gene in RNA. So it's a lot shorter because it's made from one gene. Okay, whereas DNA has many genes. Good. That's it. I realised my handwriting got a bit bad towards the end, so apologies. But I was also trying to make sure this gets done within, an hour or so, so a decent time frame. There's a lot to say about this, but I've hopefully just covered the major points. I've not gone into too much sort of extra detail, which I'm sure you'll be pleased to know. This is the main point of the news, really. If you are interested in any detail as to why, for example, we need 3 phosphate groups on an activated nucleotide, or why it's a 5' to 3' direction and all that stuff, you can obviously get in touch by email and I'm happy to answer it. But this is basically the first bit of DNA, which is the structure of DNA and RNA. So it's worth bearing in mind that RNA is just single-stranded. So it looks exactly the same. It's just one polynucleotide strand. So it would look like one of these. So RNA would just look like this, for example, where we obviously substitute the T's and U's, and it's it's ribose as opposed to deoxyribose. Cool. Hopefully that was useful, and I will see you next time.